Hello, thank you for joining today's event with Calidi Biotherapeutics, which trades on the NYSE American under the ticker CLDI. With us today, we have Andrew Jackson, Chief Financial Officer, and Antonio Santidrian, Chief Scientific Officer. We will begin with a brief presentation in a moment, and then we will answer your questions. Users may submit a question at any time by using the Q&A button at the bottom of the Zoom window. Before we begin, please allow me to read the Safe Harbor Statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical fact should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. I now turn this webinar over to Andrew and Antonio. Gentlemen, please go ahead.
Great. Thanks for having us. Thanks for the opportunity. We look forward to discussing Calidi Biotherapeutics with you. So we are a clinical stage biotechnology company that's transforming cancer with an innovative oncolytic virotherapy platform. So what makes us different from other companies is that we overcome the clinical challenge of rapid elimination of the virus by the patient's immune system. We have both a localized technology and an exciting new systemic technology. So this slide really encapsulates what makes us different. On the left, you'll see what we call a naked virus. So here's an unprotected virus that largely gets eliminated before it can reach the tumor cell by the body's immune system. On the right, you'll see our solution. So the golden orb represents a stem cell. We load our virus into the stem cell, and we then cryopreserve it. The stem cell then, and when administered, the stem cell protects the virus from the body's immune system. So not only that, but the virus actually amplifies within the stem cell. And then the stem cell has receptors, so the tumor actually embraces the stem cell, and the stem cell opens a tumor microenvironment, not knowing that the stem cell has this toxic payload. Then 48 hours at human temperature, the virus expands so much that it expands and bursts through the stem cell wall, and the full payload is then loaded into the tumor. Finally, what it does then is it elicits a response from the body's immune system to further attack the cancer. What that does is then it trains the immune system, and the body keeps that in memory. So the next time the same type of tumor presents itself, the immune system has been trained to fight it. Here we see our oncology pipeline. We have 3 platforms. The first platform we call NeuroNova. So this is for high-grade glioma or brain cancer. So we right now are about to enter a newly diagnosed high-grade glioma trial. This is with Northwest University. We have an open IND, and we expect them to start dosing this quarter. Then we're in the middle of a phase 1 trial with City of Hope for recurrent high-grade glioma. We received $12 million in CIRM funding for this, so this is largely unpaid by the company. The second platform is our Supernova platform. So this is for advanced solid tumors, including head and neck, triple-negative breast cancer, and soft tissue sarcoma. So we expect to file an IND in the first half of this year for this trial. To date, we've already received $3 million in CIRM funding for this for preclinical studies. And in general, CIRM follows their portfolio companies, so we expect to apply for another $8 million in CIRM funding later this year for this trial. Finally, our exciting new technology, which is RT-Nova. The indications for this are metastatic solid tumors and lung cancer. This is a systemic administration, and we'll get more into that in a few slides. Here we see our upcoming milestones for this year and next year. We talked about the multiple dosing trial in newly diagnosed high-grade glioma. We're expecting Northwest University to dose the first patient this quarter. Then the trial that we're in the middle of with City of Hope, we expect to have trial updates from that in the first half of this year as well. Excitingly, with Supernova, we expect the IND submission in the first half of this year and expect to dose the first patient as well by mid-year. And then finally, RT Nova, I'm pleased to say we were accepted at the AACR conference, and Antonio, Dr. Santidrian, will get into more details of RT Nova in a second. This slide really shows how the three technologies are similar and a little different. On the left is our NeuroNova platform. This has an adenovirus that gets loaded into a neuronal stem cell. In the middle is our Supernova technology. This likewise also has a stem cell and a virus, except this time it's a vaccinia virus that gets loaded into an adipose-derived mesenchymal stem cell. What you can see with both of these is it's a fairly large form factor, and we were challenged by Big Pharma to shrink the form factor and be able to administer it systemically. So on the right, you see what we've done is an individual virus through the manufacturing process gets enveloped by a human cell membrane. This cell membrane has all the receptors that a normal cell would have, which makes it invisible to the body's immune system as well. We'll go into a little bit more detail on NeuroNova. So we have, there's 3 trials. The trial on the left is a trial that's already been finished. This was completed at Northwest University and published in The Lancet. As you can see, the overall survival increased by about 50% from 12 months to 18 months. In the middle, you see our ongoing trial with City of Hope. This is a multi-dose trial for recurrent high-grade glioma. And on the right, you see the trial that we're preparing for. This is for newly diagnosed high-grade glioma, and this is also a multiple dose trial. We have an open IND, as I described, from Q4 of last year, and we expect to start dosing this quarter. This shows the trial design for the current trial at City of Hope. It's a 4-cohort trial, and we're in the 4th cohort right now. So what that means is we've had really good safety data through the first 3 cohorts. This is the trial design for the upcoming trial at Northwestern University. It's also a multi-dosing trial. As you can see, the first dose would be at biopsy. And then interestingly enough, these drugs are designed to be used with standard of care. So as you'll see, we get into radiation by the 3rd and 4th doses. And the treatment actually becomes more efficacious with radiation. It actually helps our virus be effective. Supernova, this is the technology for solid tumors. We've already completed a safety study that was very well tolerated, that was published in the Journal of Translational Medicine in 2019. And we've manufactured cGMP manufacturing for this trial, so we're prepared for that as soon as we start dosing. I've got 2 slides here that just really show visually how powerful this is. So this was from our 26-patient safety study. So here we have a patient, stage 4, metastatic head and neck sarcoma. And as you can see, after 6 months, the tumor is totally regressed. By about 2 months, it's very palpable. You can see it's very soft to the touch. And by 6 months, it's fully regressed. Here's another visual. This is a stage 4 cancer patient again. This is thyroid papillary carcinoma. It actually spread to his lymph node. And as you can see, after 3 months, we have full regression of the tumor. The trial that's coming up is going to be a 3-phase trial. The first phase is a dose escalation. Then after that, we'll be expanding it to do 10 patients from each of the 3 indications. And then the 3rd phase, we expect to be a phase 2 trial, and this could be a pivotal trial to expand on the best responding indication. So these platforms that we've talked about are all localized administration. So NeuroNova for brain cancer, and supernova for solid tumors in the body. But really, what would be super powerful, and both of those are most efficacious for these solid tumors, but most patients die from metastatic cancer. So how do you treat metastatic cancer? That's where our exciting new RT-Nova technology comes from. Let me have Dr. Santidrian walk you through RT-Nova.
Thank you so much. Thank you everybody to be here with us today. As Andrew was mentioning, the two platforms that you already saw with the two drugs that we are developing, Supernova and Neuronova. Those two are designed actually to treat, they're designed for administered on intratumoral or localized administration. Those are actually very important. As we know now in the clinic, those administration paths are the ones that we are seeing better results in the clinic. However, as Andrew was mentioning, with that type of administration, we are going to be limited. We're going to be limited to a specific type of tumors and stages. Very good, but limited. So to broad the spectrum of our pipeline and actually challenged by big pharma, we developed this platform. This platform is now in preclinical stage, and we are for the first time building a platform that is able to be administered systemically and be able to survive circulation and attack every single tumor that is disseminated into the body. So this treatment is designed to be cost effective. And also, again, I want to emphasize this. For the first time, a platform is able to survive circulation and target tumors that are dispersed in the body. So in the next slide, I want to emphasize what exactly is this platform. Now, I won't go to all the science. I'm going to do just a quick overview. But we have genetically engineered this virus so that during the manufacturing process, it's being enveloped with a human membrane. Now, this membrane will have these human receptors. That is represented on the figure on the left. And these human receptors actually are the ones that are going to be protecting the virus against the inactivation by the immune system, but also will be very important not to facilitate the targeting and the tumor homing in the tumors. And the important thing of this is that with this platform, we aim, the goal is actually to target all the tumors that are disseminated in the human body. Obviously, now when we go systemic administration, we have to engineer the virus to have a better safety profile. So we have engineered with additional mutations that actually leads the virus to only amplify in tumor tissues, not in normal tissues. In the next slide, I want to explain again why we need that protection. There is many clinicals trials and many companies they claim that they have a systemic delivery to target multiple tumors, but the reality is that they don't. Even they also show the data that as soon as they inject the virus into systemic circulation, the virus is killed, is inhibited in minutes. That's why this solution that we propose with the RT-Nova, with this extra envelope virotherapy, we can actually, for the first time, reach these tumors that systemically were not possible before. So now how this is done? So it is important what I described before. We have this new virus that is being able to be manufactured and get enveloped. But obviously, that is not enough. What we needed is also to develop new methodology, new techniques to actually allow this to happen. So our team has developed new manufacturing processes. They are revolutionary to actually keep the integrity of this membrane during manufacturing. This is a picture that actually is an example where you can have on the left a virus in blue. That's a virus that is unprotected. That's the competition. And as soon as you incubate that virus with human serum, you can see on the top right picture, that virus will be killed in minutes. And then in minutes, in 10 minutes, literally the virus, around 80%, the virus is gone. However, with our platform and our manufacturing process, we are able to keep the integrity of that membrane. And for the first time, the virus will be able to survive human serum. For that reason, it's the first time that we're going to be able to develop a systemic platform that will reach all the tumors. On the next slide, I'm gonna just put one example of some animal experiments that show that concept. We have many, but obviously we don't have time to go through all these details. But here is an example of a very complex animal model where we implanted 3 different types of tumors: neck, a lung cancer, and a melanoma. On the mouse of the right, the virus was injected only once systemically. The virus has a color, so we are going to be able to follow where the virus is going. After one single systemic administration of the envelope viruses, we can see how the virus reaches very efficiently and very selectively every single one of the tumors after systemic administration. So this is true for this animal model that has 3 tumors. We have multiple models with immune system, we also created hundreds of micrometastases in the lung. We are able to show the virus targets every one of the metastases, changes the tumor microenvironment, and also is able to kill these tumors, leading to a very efficient reduction of tumors in these animal models. So here we are. Now we have a very very important new pipeline that we are developing using this platform, using this envelope RT virus, or that actually can, for the first time again, survive and target multiple tumors or metastatic. So the first lead candidate that we are working now to finalize is going to be focusing to target metastatic lung cancer, a high unmet need that the patients needs a solution. As a platform, we are going to be able also to partner with other pharma, and that's also the objective of that platform. So we are in conversations, are seeking now and this level of development early partnerships with big pharma, with the objective to also combine with other cell therapies, or for example, be able to introduce some additional therapeutic proteins that big pharma is interested into the virus genome. So because we have a viral platform that's able to target all the tumors and efficiently kill them, but because we are able to target the tumors, we can also use these viruses to deliver something else. And this is what also some of the partners that we are in conversations may have interest, not to acquire some of these assets in the future. So again, back to Andrew. Thank you for as a conclusion. The RTNova platform delivers a breakthrough on true systemic virotherapy. We have plans to move this platform at the end of 2026 into clinical trials. This is part of our company pipeline that actually diversifies our assets. And I pause here. Andrew, please, back to you.
Great, thank you, Antonio. That concludes our formal presentation. So let me turn now to questions. Okay, question 1. How are you in terms of funding? Do you have sufficient liquidity after recent offering to meet current upcoming needs, short-term and medium-term? Yes, so I'm pleased to say in the 4th quarter of last year, we raised about $14.6 million through various public financings and use of our ATM. So we announced that we expect our cash balance at December 31 to be $9.5 million. Then also we announced in early January, we raised another $4.25 million from an offering. So this has positioned us really well on the cash side. Also in 2024, we paid off or converted $4.5 million of debt. So we've positioned the company to have a really healthy balance sheet at this stage to meet short-term and medium-term objectives. Question number 2. Which of your indications may be eligible for expedited regulatory pathways? Antonio, if you could take that one.
Yes, absolutely. So I would say very generically, almost everyone, because we are focusing on high unmet needs that the patients have. But to be specific, for example, on the Supernova program, sarcoma is a very high unmet need. The design of a trial, because it's an unmet need, allow us not only to request fast track and also other processes that allows expedite the regulatory approval, but also allow us to design the trial that we can have a phase 2 pivotal trial without the need of phase 3. That allow us to go much faster and actually have less patients involved in a phase 2, like 50 to 100 patients, but really decrease the cost and also expedites the approval. And just to finalize the question on the Artinova platform on the systemic, the metastatic lung cancer also is considered a very high unmet need. It will allow us to go through the same fast track path and expedite regulatory path for approval. Thank you.
Great, thank you. You have received grants in the past. Do you expect additional grants from the institutions you are currently partnering with? Which programs are you talking to other institutions like CERM for non-dilutive opportunities? Antonio?
Yes, in terms of grants, the programs on the clinic are fully supported also by non-dilutive money, and both of them in the GBM, but Supernova is also supported by CERM. Obviously, CERM is known to follow up with the grantees on the programs they are successful, like us. We have plans this year, as soon as we have the open IND, to apply for new grants, and we expect high chances of success. Obviously, Artinova is a revolutionary platform, and also we are planning when it's going to be the right time to actually apply for a grant. But we have also other opportunities like an NCI, SBIR, and other things that will allow us to get additional non-dilutive money to support these grants. This is program.
Great. Here's another science one. In terms of the envelope that protects the virus, is that something you can license out as a standalone or partner with other companies that could benefit from its delivery capabilities?
So the answer is yes. And this is actually part of our path. The envelope vaccinia virus, the platform is so revolutionary and is designed to be able to target systemically the tumors that allows to partner and introduce into the virus genome additional therapeutic proteins like checkpoints, other immunomodulators. The virus then will target the tumors and additionally deliver some level of gene medicines into the tumor microenvironment. This is the work that we are now doing and talking with potential partners for the future.
Great, thank you. Another one about the clinical trial. What would a phase 2 trial look like from a design and time perspective?
So linking this question to the prior one, the clinical programs that we are designing is with the vision of targeting unmet needs that allow us for a phase 2 to transform that in a phase 2 pivotal trial so that we actually can use a less number of patients to go to a BLA or approval path. The design basically will allow us to avoid a phase 3 that is very costly and very challenging for the amount of patients that are required to move forward.
Okay. Can the stock price touch $10 as predicted by analysts? I am rooting for the company to crush it and cure cancer. So great, thank you for your support. Yes, we certainly think that the stock price can reach $10, and we're all working very hard towards that. Another question, will we be able to get this slide deck after the call? So this slide deck is actually available on our website. If you go into the investor relations section, this presentation is on our website. Then there's a couple of questions that we can't answer on this call because it's non-public information. So at this stage, I think we've answered the questions that we can and that we have time allotted for.
Okay, thank you very much, Andrew and Antonio. If that is all the questions that we are able to answer, then we will wrap up the webinar at this time. But before we go, let me give everyone here ways to find out more about Calidi Biotherapeutics. You can email us at cldi@redchip.com. You may also visit RedChip's investor information page for Calidi Biotherapeutics, cldiinfo.com. There you can view and download today's investor presentation and fact sheet that RedChip has prepared for Calidi, and sign up for news alerts on Calidi. And you can now, starting this week, use our AI bot to do research on Calidi and all RedChip companies. It's called RedChat, and you can research at red.chat. Thank you again to our many participants, and thank you very much, Andrew and Antonio.
Thank you very much.
Thank you.